So, any questions about what we're doing today or tomorrow? And in case you're wondering, tomorrow after the quiz, we are going to have a lecture. We're going to start talking about the spinal cord and all the stuff associated with the spinal cord. We talked about last week. We ended up talking about how do neurons increase the strength of their signals, and the answer is they don't increase the strength of their signals. Every signal is the same, whether it's a single signal right here, a single action potential, or multiple action potential. So the signals themselves do not increase in strength or change in strength. The signals themselves change only in frequency. And so what you see here is on the left-hand side, no signal, and that's over here. The reason why there's no action potential right here is because this signal right here hasn't reached the stimulus, the threshold. Once it reaches that threshold and it exceeds that threshold, then a potential occurs, and whether it's 1, 2, or 3 potentials, those action potentials are all the same. The way that the body responds to those increased stimuli is that then they increase whatever the phenomena is. In other words, it might be sending out more neurotransmitters elsewhere in the body, might be sending out more signals from nerves, it might be more muscle contractions, more signals from glands, or more muscle contractions. So depending on where it is, the response is going to be different depending on what synapses with this action potential. So today we're going to be focused principally, revisiting a little bit of what we talked about last week, but principally what we're going to be talking about is what happens after that action potential. So this slide right here just talks about what I just said. So a stronger or stimulus results in a greater frequency, but not larger magnitude. And so now I want to talk a little bit about the differences between a continuous and a saltatory conduction, or different types of action potentials. The one that's the slowest one is the continuous action potential. That continuous action potential occurs principally in gray matter. It does not happen in the peripheral nervous system. So the reason why it doesn't happen in the peripheral nervous system is that in the peripheral nervous system you have myelinated axons. And by definition, a myelinated axon then allows for the propagation of saltatory conduction. And a saltatory conduction is where one jumps from one node to another to another. So the question could be, going back to continuous action potentials, how does the body control the direction of the signal? How does the body control the direction of the action potential? And the reason, the way that it controls is going back to that, the refractory period. Going back to the refractory period that I talked about last week, during a time when the action potential can't, that area of the axon, during that time when it can't respond to a stimulus, that prevents it from going backwards. So that, and it going backwards meaning the action potential. So in other words, the action potential goes in a characteristic fashion. It only goes from the axon hillock, down the axon, to the telodendria, to the axon terminal, and then it continues to the synapse. What happens in the synapse is what we'll talk about today, but that is always, always, always what's going to happen. It never goes reverse, it never goes in 2 directions, it only goes in 1 direction. And so this is a drawing on the right-hand side of an unmyelinated axon showing the change in the change in the ionization and the distribution of sodium potassium here, and then it jumps to the right, and while it jumps to the right, it can't jump backwards because of that refractory period, and then this area right here jumps to the right further, and it can't jump backwards because, again, that area of the axon is undergoing a refractory period. This refractory period is not very long. It's a couple of milliseconds at most, but it's still a way to prevent the dual production of dual direction of the action potential. In the previous slide, that was continuous. action potentials, and this is what I just mentioned, a saltatory action potential, one where it jumps from one node to the next. And how does this, why does this jump? Because the action potentials aren't allowed to go. There's this myelination, the myelin right here, and this occurs whether it's in the central nervous system or the peripheral nervous system. The myelin being present prevents the action potential from occurring on the axon itself. And so the exposed portion, or the nodes, are where the action potentials occur. Those exposed areas, or the nodes, are where it goes jump from one to the next to the next. In the same way that the continuous axons or continuous action potentials can't go backwards, saltatory action potentials can't go backwards either, again, because of the refractory period. When you compare the relative speed of the two neurons, that is to say, unmyelinated and a myelinated neuron, the speed or the rate of the conduction is much, much greater if it's myelinated than if it's not. In addition, tomorrow we're going to start talking about the size of nerves. Now, I'm using the term nerve and not an axon here. The size of the nerve also will increase the conduction velocity. And then finally, axons with larger diameter, individual cells with larger diameter, will have a greater velocity as well. Once it travels down that axon, whether it's a continuous or a saltatory action potential, goes to the telodendria and the terminal axon, it then gets to the synapse. And that synapse is got a couple of terms associated with it. It could be the presynaptic cell, it could be the presynaptic terminal axon, the presynaptic axon. Regardless of what it says, that term presynaptic are the portion of the cell, or the cell that transmits the signals. And when we're talking about the signals, I'm going to tell you there's two types. There's electrical and there's chemical. I'm really going to focus on the chemical today and this semester, but there is also electrical synapses. The postsynaptic cell is the receiving portion of the cell, or of these two cells, and that receiving area of the neuron, if it is indeed a neuron, that receiving area is the dendrite. And once again, there's two types of synapses I mentioned, electrical and chemical. And again, I'm going to be focusing mostly on chemical synapses. I'm going to be focusing mostly on chemical synapses, but that's not to say that electrical ones don't exist. In particular, electrical ones exist in cardiac and smooth muscle. And what they're able to do is they're able to send signals very, very rapidly and promote rhythmicity of both these two types of muscles. And it is against your self-interest, it's against your health if you end up having your ventricles and your atria contracting at the same time. So that rhythmicity, that pattern of conduction has to happen, and that's in large part due to the neurons and the electrical synapses found in cardiac muscle. Similarly, the same thing is true with smooth muscle as well. So this electrical stimulus, again, now see if any of it's familiar here. We're worried about sodium moving across the phospholipid bilayer and affecting the action potential and the membrane potential. So that's something that's familiar. And again, it's in principally cardiac and smooth muscle. Chemical synapses we've already talked about chemical synapses, although I haven't talked about that in the same way. One example of a chemical synapse is at the neuromuscular junction of the skeletal muscle. In that case, the neuromuscular junction was the neuron itself, or the axon terminal, as in my drawing is what I called it, was the presynaptic terminal, and then there's a space after that presynaptic terminal called the synaptic cleft, and then postsynaptic membrane, in particular, more specifically, the postsynaptic dendrite. If it's a postsynaptic something else, then the terminal itself, the presynaptic terminal or the presynaptic axon, by definition is a motor neuron. What am I talking about here? If the postsynaptic membrane is not a neuron, then the presynaptic terminal has to be a motor neuron. because the signal for a motor neuron ends at either a muscle or a gland. Well, not ends, but that's where the signal is supposed to go to. That's the terminal portion of it. So that's something that's important to understand. When you're talking about communication between adjacent neurons, in other words, the postsynaptic membrane in this scenario here is now a dendrite, and the presynaptic terminal is going to be sending, by definition, it's going to be sending neurotransmitters, and those neurotransmitters are going to cause action potentials. Well, it could cause action potentials from propagating down the next neuron, or we'll see in a little bit how it might not be. It might not be possible. But at that point, though, when we're talking about the communication between those two neurons, the synaptic vesicles are going to be releasing their material. Those synaptic vesicles, remember we talked already about synaptic or axonal transport. The synaptic vesicles material is going to be made in the soma or the cell body, shipped down the axon via axonal transport, and stored within the axon terminals. Those synaptic vesicles will the neurotransmitter will be released into or from the vesicle, and diffusion will cause the neurotransmitter to pass from an area of high concentration to an area of low concentration, and that's going to be across the synapse. That postsynaptic membrane, if the signal is great enough, that postsynaptic signal will then propagate down to the next membrane or the next neuron and the next action potential. Once again, that ligand-gated ion channels. What is a ligand? It's a chemical, and what chemical are we talking about? Neurotransmitters. And so here's just a picture with a whole bunch of terminology associated on the right-hand side. Here's a picture of parts of two neurons communicating. Here we have the action potential propagating down the axon terminal. You'll have voltage-gated channels that will allow for the movement of calcium into the cell, into the axon. That additional calcium into the cell causes the synaptic vesicles to release their material across the phospholipid, and via diffusion it crosses that synaptic cleft away from the phospholipid bilayer. So that exocytosis and then subsequent endocytosis is this characteristic process right here. And another term, there's more terms associated with this. There's postsynaptic terms and presynaptic membranes. Postsynaptic membrane. That postsynaptic membrane is the one that's going to be opening up in response to this signal, and when it opens up, that postsynaptic membrane then is going to allow for the neurotransmitter to enter into the cell via endocytosis. If you look right here, produces depolarizing or hyperpolarizing process graded potentials across that synaptic membrane. So we've talked about those already. And again, we're talking about the movement of sodium, of potassium, and chloride ions. Why is this NT here? I thought I changed that. So first of all, capital NT means neurotransmitter. I don't know why the book doesn't write out neurotransmitter. If you don't remove the neurotransmitters from the synapse, then that neurotransmitter will continue to send that signal, and so you need to be able to destroy or remove that from the synaptic cleft. We've already talked about this mechanism here, and that is via acetylcholinesterase. So that acetylcholinesterase is resident within the terminals there. So in this case, now this on the right-hand side, so this orientation here is just slightly different. This is the axon terminal. This is the presynaptic axon. This is the presynaptic membrane. Right here is the synaptic cleft. This is the postsynaptic membrane, and embedded in that postsynaptic membrane is this ACHE, or acetylcholinesterase. ACHE is responsible for breaking up that acetylcholine into two molecules here, choline and acetic acid. Once it's broken up, then that is no longer going to be sending signals via ACH, or sending the neurotransmitter is no longer functional. If you look here now, what happens next? So the body is sending signals in very, very rapidly, right? So there's sending signals in a matter of a couple of milliseconds at a time. And so these action potentials can happen very, very quickly. And so it's in our best interest to recycle as much as we can. The choline is imported back into the axon terminal. And then there's a number of different biochemical mechanisms to take choline, adding acetic acid in the neuron, embedding it into the synaptic vesicle, which is what I have there drawn, and then it's ready to help send signals again. So choline is the part of the ACH that's recycled. Acetic acid is just rebuilt. It's a relatively simple molecule. There are other neurotransmitters as well. There's lots of other neurotransmitters. So, for instance, there's norepinephrine. Norepinephrine is another example. It's an example of an excitatory, generally speaking, it's an excitatory neurotransmitter. That norepinephrine, the mechanism by which it breaks down is slightly different, but there's an enzyme called monoamine oxidase. That monoamine oxidase, the mechanism is the same way. It turns off the neurotransmitter by breaking it down slightly or modifying it chemically, and so as a result, that chemical modification allows the norepinephrine to be transported back into the non-functional norepinephrine to be transmitted back into the axon terminal, and then the synaptic cleft then is, the synaptic bulb is then rebuilt.
This was like more of an explanation on why another axon potential isn't sent simultaneously, right? Because like they're removing the stuff.
So what you want to do is you want to send the action potential from here to here, from right to left in this diagram, and then after the action potential is sent, you want to turn it off.
And this is what it's doing.
And this is the reason why. The previous slide and this slide, depending on what we're sending here, in this case it's norepinephrine, in the previous case it's ACH. Okay. So in both cases, if you have these two neurotransmitters floating around here, then it's going to send another signal right after the refractory period. And that's what you want to control for. You want to prevent. Okay? In terms of the neurotransmitters themselves, there's a high degree of specificity between the receptors found on the dendrites or the postsynaptic membrane and the chemicals themselves. So these neurotransmitters, I mentioned a little while ago that some of these neurotransmitters can be excitatory and others can be inhibitory, and we'll talk about what that does in a little bit. But norepinephrine is an example of an excitatory excitatory neurotransmitter, and ACH is an example of another excitatory one for muscles. But as we'll see later on, ACH is one of the most abundant neurotransmitters in the body, and ACH can be either excitatory or inhibitory depending on where we are in the body. And this diagram right here, I just blew this up. Again, just sort of summarizing everything that we've talked about over the last week or so in terms of neuron signaling, neuron structure, the receiving area of the neuron, the propagation of the action potential. This example right here, this type of action potential would be a continuous action potential. And how do I know that? Because there's no myelin on there. And then on the right-hand side, we have the release of neurotransmitters from left to right, from the axon terminal to this receiving area or the dendrite. This summary of neuron communication, this book really likes to provide a lot of textural details. I'm not going to spend a lot of time talking about this. I've already spent a lot of time. So if you look in bold or in italics rather, there's depolarization, repolarization, action potential propagation, and synaptic communication. What is synaptic communication? That's what's happening at the end of the terminal. There's many different types of neurotransmitters, and there's a slight difference between a neurotransmitter and a neuromodulator, but I'm not going to distinguish between them. So we're just going to say neurotransmitters for this class. There's many types of different examples of neurotransmitters. There's in order for a neurotransmitter to be a molecule that can be considered that, it has to be synthesized by the neuron and stored there within the synaptic vesicle. It must be stimulating exocytosis into the synaptic cleft. It must have a specific receptor in the postsynaptic membrane. If it doesn't have a postsynaptic, if all those previous criteria are present and it just goes off into the synaptic cleft and then it never gets responded to, then it's not a neurotransmitter. So it must evoke a response in the postsynaptic zone. The only way that it can evoke a response or initiate a response is if there's a specific receptor on the postsynaptic membrane or the postsynaptic dendrite. Continuing on with this sort of description of neurotransmitters here, we're going to focus particularly on the effect on the postsynaptic membrane, not so much at the mechanism or the chemical structure here. Generally speaking here, we're going to say that neurotransmitters can either have excitatory or inhibitory ways to affect neurons. So here's 5 examples of neurotransmitters. Under each of the examples there are several examples that are underlined and italicized. Those are examples that I could ask about. So for instance, biogenic amines, I'm not going to ask about indole amines or catecholamines, but I might ask about serotonin, dopamine, and norepinephrine. In particular, I want you to be able to classify where these what these neurotransmitters do and then recognize indeed that these chemicals are neurotransmitters. In terms of the names themselves, so biogenic amines, an amine is an example of a chemical with lots of nitrogen in it. You know some examples of amino acids here, 2 examples of amino acids that we've already talked about. I'm pretty sure I mentioned glutamate. I know I mentioned glycine. GABA is another example of an amino acid, so there's 3 amino acids. Purines are parts of nucleic acids, and there's nitrogenous bases that have either single or two rings here. So purines are one group of nitrogenous-based compounds that is based on nucleic acids. There's also some short chains of amino acids that can be neuropeptides. And then finally some gases. So in particular nitric oxide and CO or carbon monoxide. I used to not talk about this last one because when I started talking about this stuff was illegal. Endocannabinoids. What are those? Where would you find those?
Something to do with cannabis.
So they're cannabis related, and so that's another example of a neurotransmitter. And here's more details associated with that from the book. If you look at that last one there, endocannabinoids, important in learning and memory, nausea control, and then is generally one of the active ingredients in marijuana. All right, so now let's talk about these. And if you go back to the vocabulary list that I had for you guys in the when I was doing the drawing, on the list of terminology, there one of the last 2 terms is called EPSP and IPSP. EPSP is excitatory postsynaptic potential, and that's what I'm going to talk about right now. What happens there is that the receiving neuron, the receiving dendrite, is going to be respond in a greater fashion than it would otherwise. So that excitatory postsynaptic cell increases the action potential. It doesn't increase, it increases, or if the action potential is already being sent, it increases the frequency of the action potential. So once again, it doesn't increase the strength. So in EPSP, you have increase, and IPSP, you have decrease. And now if you look at I'm gonna go back to the graphs now. On the right-hand side, if you look at this point right here, where my pointer is, this is the resting membrane potential at the internal of the axon there. The resting membrane potential is at 70 millivolts, or negative 70 millivolts rather. And if this is an IPSP, if the chemicals there are going to reduce the likelihood of an action potential getting sent, then it's going to lower that or hyperpolarize the neuron. That hyperpolarization reduces the likelihood that that neuron then can send an action potential. If you look at the next, I'm just going backwards there in the previous slide, right where my pointer is, again, we're at negative 70 millivolts. And in this case, the EPSP then increases the likelihood that the action potential is going to get sent by bringing the resting membrane potential closer to the threshold. So that mechanism there on the right-hand side, this, you should be able to tell me whether this is excitatory example or inhibitory. And indeed, this one is excitatory, and the next one is inhibitory. So the action potential right here, in my drawings, very, very crude drawings, and so far we've only included very simple drawings of axons and dendrites. Here it gets a little bit more complicated, and if you think about it, particularly in the brain, where you have anaxonic neurons that have these appendages, these cytoplasmic extensions in every direction, it's very, very likely, indeed, it's highly likely, that all these axons there are going to be, multiple axons are going to be touching each other, and multiple axons are going to be touching the same dendrites. And so in this case here, we're talking about how does the body, remember now, the body's not increasing the speed of an action potential. It doesn't increase the strength of the action potential. The only thing it can do is increase the frequency up to a limit. So how does our body modulate or control the action potentials? And so that's called neuromodulation. So if you look at this diagram on the right-hand side, first of all, this part right here is this postsynaptic membrane, as I have it labeled. This is a presynaptic neuron, and then this is also a presynaptic neuron. This presynaptic neuron is inhibitory, it happens to be. If this presynaptic neuron then sends a signal, this part right here, where we're sending this presynaptic neuron sending a signal, is going to reduce or inhibit the release of the neurotransmitters, and as a result of inhibiting the release of neurotransmitters, it dampens or modulates the amount of neurotransmitters released. If you look in the diagram right here, this is the synaptic cleft when inhibition happens, and this is the synaptic cleft when inhibition does not happen. So the signal is decreased. Potentially, if there's more than one axon, or if there's increased frequency here, that signal could be turned off. And so the signal itself, while the action potentials could be sent down this individual axon right here, so there's this action potential is getting sent down here, the signal can get turned off, it can get turned on, it can get reduced, it can get increased. So how does, in fact, does this happen here? In other words, how do you increase or decrease the strength of these action potentials? Here, through this mechanism of neuromodulation. And there's two different ways to modulate these neurons here. There's one temporally and one spatially. So, and that term right here is called summation. And summation occurs when there's multiple signals that are, when added together, they reach that that terminal threshold, the threshold rather. If a single potential arrives, it's not going to be enough to exceed that threshold. As a result, because it's not enough, it won't send the signal. So you need multiple signals to reach that threshold, and that multiple signals can either be spatially or temporally. In this case, we have spatial summation. We have multiple inputs from here is this receiving neuron, or this main cell right here. This main cell in the soma, where my pointer is, with the nucleus and the nucleolus right here, and the axon hillock right there. That receiving area, or that trigger zone, you have multiple, in this drawing anyway, we have multiple measures of the resting membrane potential. And the resting membrane potential, when you have multiple action potentials arriving from two different areas, or more than one area, even though one of those signals is not enough to send a signal, when you add them together, it is enough, and it does exceed that threshold. And if you look on the right-hand side, that's exceeding the threshold. And there's an example of spatial summation that will, now in this case, this is excitatory. In the previous slide, I talked about inhibitory. How do I know it's excitatory? Because look at this, where my pointer is. This graph right here is exceeding the threshold, and there's a good action potential that gets propagated down. So where does that action potential happen? It happens here, and because this axon is myelinated, it then would happen here, and it would propagate down in a saltatory fashion. This is the same drawing, just different with the same consequence. Where my pointer is, right here, you end up exceeding the threshold, but the mechanism by which it's happening is different. The mechanism is when two action potentials or more, right? A single action potential arriving on the left-hand side at any of these dendrites, this one in particular, a single action potential arriving there wouldn't be sufficient to send a signal, to propagate that signal down that axon. You need more than one. If you have more than one, then it exceeds that threshold capacity, or that threshold level, and as a result, the action potential gets sent. So there's temporal and there's spatial summation, and you can have a very, very complicated temporal and spatial summation here, and indeed we do. In the brain, it's incredibly complicated. You have multiple excitatory, multiple inhibitory potentials there, sending signals to in this case, this single neuron right here, the soma, and the dendrites. There's multiple axons there that are sending these signals to that affecting this resting membrane potential, and depending on which one, the inhibitory, the excitatory one, produces the greatest one, you either have a signal getting sent in this trigger zone, or you do not. If the IPSP works too hard or is prevalent, no signal. The EPSP of the excitatory one, if they're present, or if they are more active, then the trigger zone or the action potential gets sent. So now I want to talk a little bit about the ways that things happen, in particular in our central nervous system. In our central nervous system, and I'm next on Thursday, maybe, I don't think tomorrow, but I'm definitely on Thursday, we'll talk about one mechanism called reflexes. There's different types of reflexes, and in that description here, I'm going to say there's a stimulus here, it goes here, and it either participates in the central nervous system, or it avoids completely the central nervous system, and it goes back out. Right? So that single pathway right here is a sort of an easy description of how our nervous system works. Our nervous system is a whole hell of a lot more complicated than that. Here's some examples of how they could work. I just described what's called a serial pathway. A serial pathway is you go from one thing to the next to the next. If I hit one of you on the left-hand side of your path and I cause you to move to your left here, then everyone downstream of you, if I hit Douglas and I push him to the left, everyone to the left of him would get hit. but only one person would get hit by Douglas, right? So that's called serial. Convergent pathways is when I have multiple groups or there's multiple parts of the brain working on the same problem. So as a result of these smaller, when you're synthesizing things, so your brain's working on multiple problems at the same time, and then you come to a conclusion. A divergent pathway is when you have a single signal, right? And it sends signals in multiple different ways. A divergent pathway is characteristic of lots of sensory stimuli. So those sensory stimuli can be transmitting information to the brain, and then the brain, not unconsciously, but the brain decides what to do with that information. If I go and I pull one of your hairs and you respond to me, what are you gonna do? Well, the first response is going to be pulling away, the second response is gonna be ow, and the third response will be hitting my hand away, right? So all of those responses there, you have, so the initial response is going to, from that one little individual hair that I pull. The divergent pathway, then it goes to the brain and then it sends signals, well, respond in a couple of different ways. The opposite way is, well, this reverberating circuit is not necessarily something that's that important. And well, those first three are the most important. Multiple inputs, a single output. That's an example of convergent. The one that I just described when I pulled one of your hairs was a divergent pathway, was a single sensory stimulus, multiple responses there. And there's other ways to, those are the two main ways right here. I'm going to talk more about something else. So, I thought I had a longer lecture here. All right, so, this information is pretty dense. It's going to be on the quiz tomorrow. All right, it's pretty dense. Are there any questions? Yes.
And like, what way do you think we should lay out the studying method so we could do well tomorrow? I know, like, we need to know, like, the whole, like, the steps of the communication in the neurons. So, like,
If the way that I did it for you, the first thing I did was I drew the nervous tissue, right? So the neuron with the synapse and stuff like that, right? So then so that's the first thing. The second thing I did was I talked about what happens within the neuron, within an action potential, and then what we just talked about today was much broader in the context of and what happens at the synapse. So that would be my first thing, is that if you don't know the first part, any of the subsequent things are going to get confusing. Right? In other words, if you don't know that axons send signals and dendrites receive signals, and dendrites are attached to the soma and are numerous and short, if you don't know that, then you're going to get confused when I ask questions about the EPSP or the IPSP. When you're talking, so that's the first thing. So the second thing is the reason why an action potential gets sent down a pathway, in a way, whether it's continuous or saltatory, the reason why is really important. At the end of the day, all we're talking about here is the sodium-potassium pump. The sodium-potassium pump sets that signal up. The threshold stimulus is exceeded. Once it's exceeded, then that action potential gets sent. If that threshold's not exceeded, then it doesn't get sent. So that's the second part right here, that action potential, whether it's continuous or saltatory. So the initial setup is important. And again, that initial setup is just from chapter 3, right? So we're just talking about basic cell stuff. And that's why I introduced the sodium potassium pump in the first example of active transport, because this is a super important part in this case. And then finally, this last material. Now, this last material, I'm dividing it up into 3. I'm not necessarily going to be asking it a third of like 10 questions on this material, but these are going to be more challenging questions. There's no question about that. So if you can if you are confident in the first set of questions, then the last set of questions, what we talked about today, maybe you get some of them right, but not all of them. Does that make sense?
Yeah.
So that's what I would do if I were you,
Right.
okay?
I guess my only question was, is there anything like specific we should focus on, or is it just the slideshow as a whole? Like the
So
entire
if I
chapter?
spent a lot of time talking about it in lecture and spent time and time and time again, right, then you should definitely talk about that. You should definitely know that. The idea of EPSB, if the terms that are on that list in the Bio 220 diagram, right, I have like 65 or so, 70 terms that are on the list. If they're on those lists, you should know those terms. All right.
Okay. Thank you.
So Jayana has a very important appointment right now, so I have to end class. Right?
Yep.
I knew she'd play along. No, so I am ending class right now. If you guys want to stick around.
Oh my. How could you?
So if you want, I can go over more details about this, but
No, I'm just kidding. It's okay.
right? It's not, the details are the details, and I've already gone through them.
Look at me, bro.
If you need me to go through them again, you guys can stick around and I'll go through them again. But otherwise, I'm not going to present anything new. I think this is a reasonably challenging chapter to have a quiz on tomorrow.